thanks a lot. That's very generous, Bill. And it's really great to be back here and see so many people come to talk and see some friends that I haven't seen in a really long time. Okay, so as Bill said, when I was here, I was, I got to do a project in Bill's lab and making some, I think, fairly simple compounds. And I got to work with Tom. And I think, as Bill said, the thing was that I got to kind of get in there and use the NMR instruments. And that seemed pretty fun to me, and it seemed like you could do quite a few things with NMR. It kind of really stood out to me as a really useful method. And so then I went off to graduate school, and I happened to go into an apartment where there was a really great bio-NMR lab. And there was actually a couple UPS students in that lab already. And that kind of, I think, helped me get into the lab. A lot of people wanted to go into that lab. And I joined that lab, and I thought that I didn't know a lot about protein NMR at the time, but I thought that I think, if anything, could reveal some detailed information about proteins that was NMR. So, and then Bill kind of told you my history. I got to go into some other great NMR labs, and so now I'm basically a biochemist, and I use NMR as one of our major tools. And so today I'll tell you a little bit about how we're using it to study the particular proteins that I'm interested in, which are signaling proteins of this family of proteins called TGF-beta. So these proteins are not that big as far as proteins go. They're homodimers. The monomers are 12.5 kilodaltons each, and they're covalent homodimers with a disulfide bond linking the two monomers together. These proteins signal, like most signaling proteins, by, so this is the schematic of the TGF-beta dimer, by binding and bringing together two homologous but functionally distinct single-spanning transmembrane receptors known as the type 1 and type 2 receptors, TBR1 and TBR2. So these receptors have relatively small, but actually very heavily disulfide bonded extracellular domains, and single-spanning transmembrane domains, and then cytoplasmic kinase domains. So when TGF-beta binds the receptors and brings them together into a symmetric heterotetramer with two type 2 receptors, two type 1, that induces a transphosphorylation mechanism where the constitutively active type 2 receptor, TBR2, phosphorylates some very highly conserved serines in a regulatory domain of the type 1 receptor known as the GS box. When these serines get phosphorylated, that relieves the inhibition of the type 1 receptor, and in turn phosphorylates the cytoplasmic effectors of the pathway, which are known as SMAD proteins. And these SMAD proteins are normally cytoplasmic, but when they get phosphorylated, they translocate to the nucleus, and they regulate gene transcription. And I'm not really going to get too much into the biology of these proteins. That's many more lectures on its own. Just suffice it to say they're important in regulating cell growth, regulating the immune system, regulating many different aspects of development. They're really important proteins. So, and what we see is that TGF-beta is actually part of a representative of a large superfamily of signaling proteins known as the TGF-beta superfamily. There's 3 of these proteins in worms, 7 in flies, and 35 in humans. So as organisms have become more complex, the proteins of this family have diversified. So this is the phylogenetic tree showing the sequence similarity for the different proteins of this signaling superfamily. The TGF-betas are here. There are 3 of them. And the reason I wanted to show you this is because what we think is that the TGF-betas are probably among the most recently evolved members of the family. Whereas these proteins over here, of which there are a number, these are called them BMPs, they're called bone morphogenetic proteins. The reason that we think that these are among the most recently evolved, these are the ancestors of the family, is because we find homologs of the TGF-betas only in humans and other higher vertebrates, but not flies and not worms. The BMPs, in contrast, we find not only in vertebrates, but also in lower organisms, including both flies and worms. These ligands in the middle, these are the so-called activin and inhibins group. You find them in humans and flies, but not worms. So it seems pretty clear that these are the ancestors. These are among the most recently evolved members of the family. And I'll come back to that, because I think our structural work speaks to what's happened as the family has diversified from these ancestors to these more recently evolved members of the family. So there's a number of structures available of these protein, of the signaling ligands themselves. Well, a lot of them are determined by crystallography. Although this one right here is actually one that I worked on when I was at the NIH. It's actually how I got interested in working on TGF-beta. We actually determined this structure using NMR spectroscopy. The point I wanted to make here is that these structures are all similar. If you look at them at least at this level of these cartoons, you can see that they're all pretty much indistinguishable from one another. In the superfamily, there are a total of 7. So in vertebrates, there are 7 known type 1 receptors and 5 known type 2 receptors. And there's structures now available for a number of those. And these are just overlays of TGF-beta type 1 and BMP type 1 receptor. You can see they're, again, at least the level of this cartoon, the same overall fold. The same for the type 2 receptor. This is an overlay of the TGF-beta type 2 receptor and the BMP type 2 receptor. And you can also see that actually the type 1 receptors are the same as the type 2 receptors, as shown here by an overlay of the TGF-beta type 1 and type 2 receptors. So in this signaling superfamily, there's actually only one receptor fold. This is called a 3-finger toxin fold. So all the receptors, whether they're type 1 or type 2, share that same overall fold. This is an overlay of the kinases. Those are from TGF-beta and the BMP type 2 receptor. You can see those are also similar. So if this signaling superfamily has diversified, presumably it's diversified to regulate the increasing complexity of the organisms as they become more complex. There's just more things to control. It's really kind of hard to understand, at least at the level of these cartoons, because all these signaling ligands look the same, all these receptors look the same. So really what I want to focus on in my talk today, and what I've been focusing on in my research in my own lab for the past 15 years, has been trying to understand, well, how do you actually achieve this diversity in light of this family of structurally similar ligands and receptors. So some clues as to how these ligands might achieve their specificity in inducing their biological activities came from some what I consider fairly crude cell-based studies. So these cell-based studies were done by ionating the ligands. So either a BMP or these called homologs of BMPs called GDFs. TGF-betas are activin. You ionate these so they're radioactive, and you throw them at cells, and then you add a chemical crosslinker. So this has reactive groups, maleimide groups, on like a dumbbell with a linker in between, and these will react with both the ionated ligand and then with a receptor, and you can make a cross-link complex. So using this kind of crude methodology, and then you can run these things out on gels, and you can see which ligands are binding to which receptors. So using that kind of crude methodology, it was found that BMPs and GDFs bind to multiple type 1 receptors and multiple type 2 receptors. The TGF-betas and these fairly closely related ligands, the inhibin activins, in contrast, were found to be much more restricted in their receptor binding binding only one type 1 receptor and one type 2, or two type 2 receptors in the case of activin. So that's one difference. The TGF-betas and activins seem to be more restricted in their receptor binding compared to BMPs. Then another thing was that, and when you do these experiments, you can also ask the question, what effect does ligand binding to one receptor have on the binding to the other receptor? So what was seen for TGF-beta was that TGF-betas would bind very well to their type 2 receptor, but not their type 1. And they would only then in turn cross-link to their type 1 receptor after they had first bound to, after the ligand had first bound to the type 2 receptor. So it seemed that somehow TGF-betas and activins would bind their type 2 receptor, and then somehow that would potentiate the binding of the type 1. Whereas for BMPs and GDS, it was not really seen to be that way. Instead, some BMPs would cross-link to their type 1, and that would have only marginal effect on the cross-linking to the type 2, whereas some other BMPs would bind well to their type 2, and that would only have marginal effect on their type 1. So that's kind of, doesn't prove anything, but it kind of suggested that mechanistically there might be differences in the way these different subfamilies of ligands bind and assemble their receptors into active signaling complexes. So I became interested in that. As I mentioned, my lab kind of got into this by working on the structure of TGF-beta1 about 15 years ago. So I decided we're going to work on these receptors. There was really nothing known about these receptors at the time, so that's what we set out to do. We started out working on, most of my work has been on the TGF-beta receptors, and we started working on the TGF-beta type 2 receptor, because that's the receptor that binds TGF-beta with high affinity independent of the other receptor, the type 1 receptor. So, first thing you want to do this is you've got to get pure protein. Okay, so the way we do that is we make this in bacteria. We put it downstream of a strong phage promoter, make a plasmid construct of that in bacteria. Bacteria make the protein. As I mentioned, these receptors have a lot of cysteine residues, a lot of disulfide bonds. So as you might expect, when you express this human protein that has a lot of disulfide bonds in bacteria, it doesn't fold, it doesn't make the proper disulfide bonds, and it forms an insoluble aggregate known as an inclusion body. So you break the cells open, you centrifuge, you take the pellet, which has the insoluble inclusion bodies, you then denature that in very high concentrations of urea, like 8 molar urea, and then you reduce. So now you have an unfolded polypeptide with the cysteines in the reduced state. And then you, by various methods, play around with it. You basically then transfer that to a non-denaturing environment. So basically diluting from this reduced urea-containing solution into a non-denaturing buffer. So something without urea and without reducing agents. And then you fold in the presence of some reduced and oxidized glutathione, and that promotes a disulfide exchange. And then you take your protein that hasn't precipitated when you folded it, and what remains soluble, concentrate it down, and then purify it, and see what you got. So that's what we did, and we actually got really, this protein folded very well. Most of it, when you fold this protein, most of it stays in solution, unlike a lot of proteins when you fold them, which precipitate. So this protein stayed in solution. We purified it, got a single peak on the HPLC, and then most people would do the functional assay. But actually, my training was as an NMR spectroscopist. So actually, what we did was we just made an NMR sample and then looked at it in the NMR instrument. So the way we do this is we make, when we express the protein in bacteria, instead of making just growing on regular growth medium, we'll make a minimal growth medium with N15 ammonium chloride. That way the protein gets N15 at all the nitrogen sites in the protein. And we're particularly interested in the backbone amide. And the reason for that is if you label the protein with N15 and look at the backbone amide, you basically get one signal for all the amino acid residues except for prolines, and you should get basically a spectrum. If you run an NMR spectrum like this, where you have correlating the amide proton to the amide nitrogen in a 2D, you should basically get one spot for every amino acid in the protein. And when we do that, that's basically what we got. And so if you count the number of signals there, it correlates very closely with the number of amino acids in the protein. And the other thing about this NMR spectrum, and it's really akin to the kinds of things that you do with small molecule NMR, is that we saw that the amide signals were very well spread out. Okay, so if you have, if you just look at a model amide in a peptide, like just a random tetrapeptide, and you look at all the amides, you'll see, no matter what peptide you look at, will come between about 7.9 and 8.5 in the proton dimension. Okay. Also, you can see the same thing if you take a protein, you put it in denaturing, like 8 molar urea, then the spectrum will collapse. You'll find all of your amides in that narrow band. And the reason is because when the protein folds, all the amides now actually, some are deshielded, some are shielded, and that increases the spread of the signals. So typically in a folded protein, you'll see the amides spread out between about 6 and 10 ppm. And so that's what we see here. It's a really nicely dispersed spectrum with the right number of spots. It's just by looking at that alone, you can't prove that the protein is in its native conformation, but it sure seems to have all the attributes that are consistent with it being in a native single conformation. So, of course, then you have to actually do the functional assays. Does it actually bind TGF-beta like it should according to the cell-based studies? Here we're just doing in-vitro binding. So we're binding purified TBR2 extracellular domain to purified TGF-beta3 dimer. And this is a very simple experiment, but I really like it because it's so simple. Basically what you're doing is you're just titrating increasing molar ratio of the TBR2, purified TBR2 ectomane versus a constant molar amount of the TGF-beta dimer. So here, if you run the receptor by itself, it runs quite rapidly because it's actually highly negatively charged at the pH of this gel. The TGF-beta is actually has the opposite characteristic, so you don't, when you load it on the gel, you don't see it by itself. But then if you add increasing amounts of the receptor to TGF-beta, you see this complex band appear. And what's really cool about this is you can see an increase in intensity up to the point where you have 2 equivalents of R2 per each TGF-beta dimer. And then when you go beyond that, you then begin to see excess TBR2. So this was really nice because it's kind of exactly what we expected. This is supposed to be the high affinity receptor for TGF-beta, and we'd expect it to bind, because TGF-beta is a dimer, we'd expect the receptor to bind with 2-to-1 stoichiometry. So that's what it seems to be showing. So then we wanted to be a little more quantitative about this. So what we do very extensively in our lab is to use a method called surface plasmon resonance to measure the binding and quantify the binding constants as well as the rates. And the way this instrument works is you have a gold surface with a dextran layer on it, and you can covalently couple proteins to that using carbodiimic coupling. So in our particular experiments, we typically will carbodiimic couple TGF-beta dimers to this gold dextran surface, and then this instrument has a microfluidic system where you can inject very tiny volumes of receptor. You can vary the concentration of receptor that you inject. And the way this instrument's very sensitive. So basically what, this is actually a very gross oversimplification of the physics. I hope Professor Thorndike is not here. But basically what happens is when you have a binding here, and the mass density on the surface is increased, that basically changes the refractive index of the surface. And then you can detect that using an optical detection system. So this is really sensitive, and it's a really handy way, because as I said, you not only get the binding constant, but you also get the on and off rate. So this is the kind of data that you get using SPR. So here we have TGF-beta3 immobilized on the sensor chip surface. We inject increasing concentrations of the purified type 2 receptor, And as you increase the concentration here from 0 to 1 micromolar, you get an increasing response. This corresponding to this bar is when the receptor is being injected. At this point, you stop the injection, and then the receptor dissociates. So this is the association. This is the dissociation. So you can fit those curves and get the on and off breaks. You can also look at the maximum amplitude of the response as a function of injected receptor concentration, and fit that to a standard binding equation, we get the KD. So if we do this, looking at the maximal response as a function of receptor concentration, we get KD of 0.22 micromolar. So this receptor seems to be binding pretty well. So then so that's good. That's only half the story, though. We need the type 1 receptor as well. So if we want to understand really how TGF-beta binds its receptors, so we went after that using similar methodology that I told you before. And then this time when we put it in the NMR machine, it didn't look so great. So in this case, we saw most of the backbone amides were within what we call the random coil limit. There were very few signals outside the random coil limit. These signals right here, I skipped over that before, but you also get signals in this experiment from the side chain amide groups of asparagine and glutamine. So that's what these signals are. So these are not backbone amides outside of the random coil limit. Those are the side chain amides, and that's where they typically fall. So there's very few signals in this NMR spectrum. Most of them are in the random coil region of the spectrum. So this shows probably that this protein is either in a single conformation that's non-native, where it's essentially misfolded, or just multiple non-native conformations. You can't distinguish that. But in any case, it's not good. It doesn't look like the other one. It's probably not native protein. Unfortunately, we couldn't verify that for sure using a binding assay because we had no binding assay for this protein. And we also, because we're making a human protein in bacteria with a lot of disulfides, with TBR2, it didn't fold. That's kind of what you expect. You don't really expect to get native properly folded protein out when you express these proteins in bacteria. So we then switched to eukaryotic cell expression systems that have biochemical machinery to fold these proteins, or they should have the machinery to fold these proteins. They have chaperones, they have proteins that modulate the redox potential and so forth. But unfortunately, we had no success in obtaining, expressing this TBR1 ectodenate either in insect cells or cultured eukaryotic cells like CHO cells or HEC293 cells. So we're kind of stuck. We have no assay. The bacterial protein looks bad. We weren't able to get it from eukaryotic cells. So we just kind of scratched our heads and thought, maybe actually in the E. coli refolding mixture of this protein, there is actually some native protein, but a minor fraction. And if we could find out if there was some native protein in there, we could then hopefully separate out the native protein from the non-native protein using HPLC. So, and then we thought, well, the thing about this type 1 receptor is that its kind of defining characteristic is it binds well to TGF-beta when it's bound to the type 2 receptor, but not bound, but by itself it does not bind TGF-beta well. So we went back and what we did was we basically asked the question, if we take our folding mixture, which appears to be heterogeneous of this TBR1 protein, and then add that to the purified TBR2 TGF-beta complex, can we see any component of that folding mixture bind to the purified TBR2 TGF-beta complex? So, and again, we turn to that using this very simple native gel assay. So, basically, what I want to point your attention to here is our TBR1 by itself. You kind of get this kind of crummy-looking smear. If you add this crummy-looking smear to TBR2, you just see the crummy-looking smear in the TBR2. If you add the crummy-looking smear to TGF-beta, you just see the crummy-looking smear and no TGF-beta like you normally see. But then, if you take the So here we have the TBR2 TGF-beta complex. We know that that's a native TBR2 bound to native TGF-beta, and it's the 2-to-1 complex. So now what we're doing here is we're adding that crummy smear to this purified TBR2 TGF-beta complex. And then what you see when you do that is this other band show up. Okay? So now we're adding about 100-fold excess of this smear, molar excess of this smear protein relative to this complex, yet only about half of it is consumed to make this other species. And that other species is presumably the ternary complex, where you have this with natively folded TBR1 also bound. And so to prove that, what we did was we took this band, cut it out of the native gel, and then put it on a denaturing gel, an SDS gel. And here you can see when you do that, this is this complex here, we call BC, the binary complex. This one here is the ternary complex. Here in the binary complex you see TBR2 and the TGF-beta dimer, but in the ternary complex you see TBR1, TBR2, and the TGF-beta dimer. So this native gel assay, even though it's very qualitative, seems to be suggesting that there is in fact native TBR1 that's interfolding mixture that's capable of binding the TBR2 TGF-beta binary complex. So that was really actually encouraging to us. This was actually our first assay for this protein. So what we did was then we took our folding mixture, which we knew was heterogeneous, fractionated that on high-resolution ion exchange columns, and then subsequently on C18 reverse phase columns. There were clearly many species in there, but using this assay we were able to identify the native protein, and then when we made up an NMR sample of that, put it in the NMR spectrometer, then we were much happier. Okay? You see we got a really nice spectrum here. Here we have very nicely defined signals. The number of signals, again, corresponds to the number of amino acids in the protein. And again, we have many signals that now, backbone amide signals, that lie outside the random coil region of the spectrum. So this was consistent, with what we were seeing in the native gel, that we were pretty confident at this point that we had identified and purified the homogeneity, the natively folded TBR1 extracellular domain. So this is now just going back with the purified TBR1 extracellular domain, just repeating what I showed you before. Here's the binary complex, and if we add our purified TBR1 ectodomain to the purified binary complex, we get a quantitative shift of all the binary complex. The ternary complex, we do a titration, where we're titrating a fixed amount of TBR2 TGF-beta binary complex with increasing concentrations of TBR1. We see the binary complex go away and the ternary complex increase, and then we start seeing excess TBR1 when we've gone beyond 2 equivalents of TBR1. So this pretty much proved that we had isolated native TBR1 that bound the TBR2 TGF-beta complex with a stoichiometry of 2-to-1. Okay, and then we want to again quantify this using SPR, basically the same method that I told you before, but with one trick. So if we bind here again, we have TGF-beta on the SPR sensor surface. We inject increasing concentrations of TBR1. Here we're going from 1 to 70 micromolar. You hardly see much of a response. If you do a similar kind of experiments with immobilized TBR1 and inject increasing concentrations of TBR2, here we're going up to 100 micromolar. You see no response. So TBR1 binds weakly to TGF-beta. TBR1 binds weakly to TBR2. But here in this experiment, what we're doing is, again, we have TGF-beta on the sensor surface, but now in the SPR buffer, we put a saturating concentrations of TBR2, of native TBR2. And that buffer is always going over the TGF-beta on the chip surface. So, and then we inject on top of that our purified TBR1. And so now we have TBR1 binding to essentially TBR2 TGF-beta complex on the chip surface, not TGF-beta alone. And when we do that, injecting only between 0 and 1 micromolar, we see a very robust response. So, and that's in contrast to what we saw here. Here we're just injecting TBR1, same thing that we're doing here, But the only difference is that this TGF-beta is bound to TBR2. This TGF-beta is not. So the presence of TBR2 bound to TGF-beta greatly potentiates the binding of TBR1. And that's, again, fully consistent with what had been seen in the cell-based studies, the crude cell-based studies using the cross-linker. If we quantify the dissociation constants, in the absence of TBR2, TBR1 binds extremely weakly. This is a very approximate KD of about 0.5 millimolar. In the presence of saturating concentration of TBR2, TBR1 binds with a KD of 0.37 micromole. So if you believe those KDs, and I wouldn't, especially this one here, this is very approximate, but nevertheless, it's clear that TBR2 is very strongly potentiating the binding of TBR1, probably by a factor of 1,000, or maybe even more than that. So it's a really, this is not a subtle effect, it's a very pronounced effect. So now we had the receptors in hand, and we were interested in trying to look at this at the molecular level, get the molecular structure of TGF-beta bound to its receptors, and then the idea is we ultimately want to try to compare that to BMPs bound to their receptors. So the way we went about that was by adding an excess of the receptors to purified TGF-beta dimer, and then loading that on a gel filtration column. And so these are our profiles. We ran the TBR2 TGF-beta complex as a control. And so here we have the void volume. So this is the high molecular weight aggregate coming off. Remember, on a gel filtration column, the higher molecular weight material comes off first, and the lower molecular weight material comes off last. So here we have aggregate coming off. Here we have like contaminant UV absorbing small molecules in our buffer coming off. And then in between we have our proteins. And here's our ternary complex. So this is TGF-beta bound to TBR1 and TBR2. This is the binary complex that comes off a little bit later, which was what you'd expect because that TGF-beta is bound to TBR2, but not TBR1, so it has a little bit lower mass, lower by about 20 kilodaltons. And then here are our unbound receptors. And you can note that by running these on SDS gel. And so this is a native gel of our isolated ternary complex. This is our isolated ternary complex. And then right next to that we have what we call assembled ternary complex. That's just ternary complex that we assembled from the purified components. So this indeed is a native complex where all three proteins are binding to one another, forming a very stable, non-dissociating complex that we can isolate over the course of hours on this gel filtration column. So that's really good. We have this really nice, stable, purified complex. And that's exactly what you want if you want to try to crystallize it, which is what we did, because this complex is far too big for NMR. This complex is about 100 kilodaltons in size. So doing NMR on that, even though NMR has come a really long way, that would be really expensive and hard to get the structure of that kind of complex using NMR. So for this, we turn to crystallography, X-ray crystallography. So we make a concentrated solution of this purified complex, and then mix it with various precipitants. Just empirically screen for crystal formation, and we got lucky, got this pretty big crystal. If you take one of these crystals and dissolve it and run on SDS gel, you see all three proteins are in there. So this is a crystalline lattice of this protein complex. So then we collect diffraction data. We do that, in this case, at the advanced photon source. So that's the synchrotron, X-ray synchrotron radiation at Argonne in Chicago. And we collect all the diffraction data, and then we were ultimately able to solve the structure of this complex to 3 angstroms resolution. Okay. And this is what the structure looks like. So here we have now the TGF-beta dimer in the middle. Here in green, on either end of the TGF-beta dimer, we have TBR2. And then nestled between the TBR2 and TGF-beta, we have the TBR1. So this is so for each TGF-beta dimer, you have 2 TBR2s and 2 TBR1s. And what's interesting, and I think really stood out about this complex, was that you actually see an extensive interface between the type 1 receptor and the type 2 receptor. You can see that here in the surface representation. So here you have an extensive packing between the type 2 receptor and the type 1. This turns out is actually the N-terminal tail of TBR2. So this is just a linear peptide of about 6, corresponds to about 6 residues that's highly extended, and that packs against the surface of TBR1. And so that was of interest to us because the thing about TGF-beta that really made it distinct, that we saw in vitro, that had been seen in cells, is that TGF-beta binds its receptors in a very ordered manner. First by binding the type 2 receptor, and then, and only then, by effectively recruiting the type 1 receptor into the complex. And so based on the structure, we hypothesized the reason for that was that in effect TBR1's binding site is not fully formed until the type 2 receptor binds. It really, we were hypothesizing that the type 1 receptor binding is really, it depends on these interactions, but it also depends on that interaction here. And the reason we were hypothesizing that TBR1 doesn't bind well in the absence of type 2 receptor is that this region is absent. So we want to test that. We don't want to just look at the structure and say that it has to be that way. We actually want to prove that. So, so this is just an expansion showing this interaction here. So here we have the TBR2. Here we have in background the TGF-beta in blue, and then the type 1 receptor in yellow. So you basically have this N-terminal tail of TBR2 lying on the surface of TBR1. And what we see is 2 very highly conserved hydrophobic residues, a valine and a phenylalanine, that pack into a hydrophobic pocket on the surface of TBR1. So we hypothesized if we remove this tail, make a truncation where that tail is removed, or we mutate those residues, that's going to not affect TBR2's ability to bind TGF-beta, but it will affect TBR2's ability to bind and recruit TBR1 into the complex. So, so that's what we did. We made a number of TBR2 variants. So here's where the tail's removed. Here's where we're making mutations, mutating the phenylalanine. And basically what we see is, regardless of when we mess around with this tail, it has very little effect on TBR2's ability to bind TGF-beta. But if we remove that tail entirely, whack it off right here, you see that the KD for TBR1 recruitment goes up nearly 100-fold. So, so our biochemical data seem to support what we proposed based on our structural analysis. And I should say, I'm not going to show it here, but we also looked at this in the context of the receptors on the cell surface by looking at the mutant receptors. In the context of the cell surface, we saw the same result. So this is all very satisfying because we basically, by purifying these proteins and then getting the structure using crystallography, we basically felt like we had come to identify the underlying reason for why TGF-beta assembles its receptors in such a stepwise manner. So now, while we were working on TGF-beta, and I should tell you what I just told you, it took about 10 years, groups in other labs, one group in particular at the Salk Institute in La Jolla, and another group in Germany, in Würzburg, they were working on BMPs. And they, after toiling for a similar number of years, came to a similar structure, or a similar type of structure for, in this case, BMP2, down to the BMP type 2 receptor, one of the BMP type 2 receptors, and one of the BMP type 1 receptors. So this is the type 2, the type 1. But what you can see is that that structure, the structure of that complex, the way the BMPs binds its type 1 and type 2 receptors is entirely distinct from the way TGF-beta binds and assembles its receptors. So I'm not going to go through in detail, but basically the interfaces that both the receptor uses to bind the ligand, and as well that the ligand uses to bind the receptor, are entirely different between these two protein complexes. And the consequence of that is that if you rotate this molecule around, there's large gaps between each of the 4 receptors in that complex. In the BMP receptor complex, there's no receptor-receptor contact, and that's because the receptors are positioned on the ligand totally differently than they are in the TGF-beta receptor complex. So what seems to have happened over the course of evolution is that it started out this way, but then mutations accumulated, other modifications occurred that caused the receptors to bind the ligands differently, in a entirely different mode, and in particular to enable this, and such that enabled this receptor-receptor contact that led to this pronounced stepwise manner of assembly for TGF-beta, but not for BMPs. So we think that that's a really defining and important evolutionary modification that has occurred that essentially allows TGF-beta to discriminate its receptors from BMPs and its receptors. So now I just want to mention one more thing about this, and that relates to this N-terminal tail. Because basically what I'm saying is that I think that this N-terminal tail of TBR2 is really important for the way TGF-beta binds and assembles its receptors into a complex. So we were interested in that in particular because this N-terminal tail, as I showed you in our structure, is highly extended. So we thought that probably it's actually not that way by itself when the type 2 receptor is not bound to TGF-beta, that it probably undergoes this ordering reaction as the complex is assembled. So we wanted to look at that, and to look at that we used NMR. So here, this is the 2D proton-nitrogen correlation spectrum of TBR2 that I showed you before. The thing about this is, even though it's a really nice spectrum, we don't know which amino acid is giving rise to which of those spots. And if we want to say something specifically about the tail, we need to know which amino acid is giving rise to which spot. So we need to basically assign our spectrum. And the way we do that using, the way that's typically done is to collect 2 different shift correlation, 3D shift correlation spectra. So these are proton, carbon, nitrogen 3D spectra, and you simultaneously correlate the chemical shifts of, in one experiment, the amide proton, the amide nitrogen, the C alpha or C beta, or CO, or some combination of those 3, all in one experiment. So you have on one axis the amide proton, on the other the amide nitrogen, and on the third carbon axis some combination of these carbons here. And then you run another experiment that's similar but different. So you have, again, the amide nitrogen pair, but in this case you go backwards, correlating to the CO and the C alpha, and in some cases the CB. So again, another 3D experiment, but in this case generating what we call an inter-residue correlation between the carbons and the amide, as opposed to this other experiment where we have an intra-residue correlation of the C alpha to its own amide. So you collect these 2 separate 3D data sets on a sample, on a protein sample that's both now N15 labeled and carbon 13 labeled. And what you do is you then look down for each amide. You look down in the carbon, along the carbon axis, and the way you do that is you basically just pick an amide, for example, from this experiment. You know that according to the way this pulse sequence works, it's correlating this amide to its own C alpha. So what you do then is you go to the blue data set and you look for a different amide that correlates to that same exact carbon chemical shift. And when you find that, you know that this amide that you picked from the first experiment, the intra-residue experiment, must precede the amide that you found that matched with that from the other experiment. And so doing that procedure, and that's what I'm showing here. So here, if you look at this glutamate 55, for example, this is the intra-residue correlation. So if you look at that guy, and so this is its intra-residue correlation. If you then go to the next residue and you look to its inter-residue correlation, you see two peaks, one for the beta and one for the alpha, that match exactly the one for the inter-residue correlation for this residue. So you know, based on that, that these two residues have to be next to one another. And then similar going in the other direction. So doing this, by matching up these carbon shifts, you can essentially walk down the protein backbone, either in the forward direction or the reverse direction, and essentially identify all the, assign all the amino acids in the protein. And this actually works pretty well, especially if one uses not just the C-alpha carbon shifts alone, but also the carbonyl and C-beta, because there's often cases where you have two residues that have, or more than two residues that have similar carbon chemical shifts. So there's some degeneracy, but you can overcome that by looking at multiple carbon nuclei simultaneously. So anyway, doing that, we were able to assign all the backbone amides in TBR2, and that's shown here. So now the spots come with nice labels, so you know which amino acid they're coming from. And then you can kind of do the experiments that I was talking about, which is asking the question, does the TBR2 N-terminal tail, is that disordered when the receptor is not bound, or is it actually pre-ordered? So to look at that, so now we're using those assignments, and to look at whether something is ordered or not, what we do is we measure different NMR relaxation times. And the reason for that is because the NMR relaxation times are sensitive to the molecular dynamics. In this case, there's two kinds of dynamics. There's the overall tumbling of the protein. That's one kind of motion that the amides experience. Another motion that the amides experience is just internal motion. Within the particle, how flexible are they? And so these different relaxation times, so we're looking at the nitrogen T1, the nitrogen T2, and the nitrogen proton heteronuclear NOE. These are sensitive to motions on different time scales, but basically one thing that you can clearly see is that on the tail of the protein here, on the N-terminal tail, but as well the C-terminal tail, you have a precipitous drop in heteronuclear NOE. And you also have a significant increase in the T2. So that's consistent with this region of the protein being essentially approaching a random coil. So this part of the protein we know is all structured, and then because these relaxation times are changing, that tells us that these regions are highly flexible. So you can basically take this relaxation data and do some modeling, and then arrive at what's called an order parameter. This parameter goes between 0 and 1. 1 means that that amide is perfectly rigid on the time scale that you're probing it at, which in this case is on the nanosecond to picosecond time scale. So what you see is that across the structured part of the protein, where we have all the beta strands that are hydrogen bonded to one another, you see the backbone is essentially kind of uniformly rigid. But then on the N-terminal tail, as well as the C-terminal tail, you can see it becomes highly flexible. The order parameter essentially goes to 0. That means that this region is approaching a random coil. So this NMR data tells us that if you just look at TBR2 alone, the N-terminal tail is disordered. That region that was ordered in the complex structure is actually disordered in the receptor by itself. We also wanted to look at TBR2 when it's bound to TGF-beta but doesn't have TBR1 there. Is it also disordered in that context? So again, doing NMR on the TBR2 TGF-beta complex is a little bit challenging. That's about 60 kilodaltons, which is getting to be pretty challenging. So what we did was we crystallized this TBR2 TGF-beta complex. So in this case, there's no TBR1 there. We're able to get nice crystals. We were able to get the structure again. And when we looked at the structure, we saw that here, again, we have exactly what I showed you before. There's no TBR1 because it wasn't there. But in addition, even though this part of TBR2 was included in what we crystallized, in spite of that, there was no interval electron density for that N-terminal tail. So that's consistent with what we see here in the NMR of the unliganded receptor. So it seems that when TBR2 is bound to TGF-beta, well, either in TBR2 alone or when TBR2 is bound to TGF-beta, this N-terminal tail is disordered, and it only becomes ordered when TBR1 binds. So this is an example of what we call in biochemistry induced fit binding. So the TBR1 binds into this cleft, TBR2 binds, creates a cleft for TBR1 to bind. Initially, when the TBR1 binds, presumably this tail is disordered, but when the TBR1 binds in there, presumably then that tail undergoes a disordered order transition and essentially forms contacts which then lock the receptor into the complex. And presumably this is actually important for the specificity of TGF-beta for its receptors, because if you have, like, for example, a non-cognate BMP type 1 receptor enter into that cleft, this tail will attempt to form those complexes or form those contacts, presumably because those surface residues are different. It doesn't have the pocket that TBR1 has, that receptor, even though it binds to the cleft, it then dissociates relatively rapidly because it doesn't form these stabilizing contacts that are formed through this ordering of the TBR2 N-terminal tail. So that's so again, we think that this receptor-receptor interaction, the TGF-beta receptor complex, is very important for the specificity of TGF-beta for its receptors. So basically, what we think has happened here is we had this. These are again the ancestors of this signaling superfamily. These receptors bind the BMP independently. When they bind, they don't contact one another or stabilize one another. These receptors, I don't think I mentioned, but these receptors that bind BMPs activate what are called SMADs 1, 5, and 8. Whereas the TGF-beta, all these ligands over here bind type 1 receptors that activate SMADs 2 and 3. So what we think has happened is that as this protein signaling superfamily has diversified, that there was a schism where the binding mode changed from this more ancestral type to this more interdependent manner of binding. And in so doing, that was coupled with activation of a distinct group of SMAD proteins that actually target different genes than these SMAD proteins. So basically what we think we've uncovered is at least one mechanism by which the TGF-beta superfamily has diversified its function. Essentially, through these new binding modes, it's essentially allowed the activities of these ligands to be, these more ancestral ligands of the superfamily, to be segregated from these more recently evolved members of the superfamily. Okay, I have more, but now it's 52 minutes, so I'm going to skip over that. Let me just briefly tell you, I just already summarized that. Let me just briefly tell you what I was going to tell you in this latter part of the talk. I will not go through the data. So we think we've uncovered at least one mechanism that's kind of segregated, that's evolved to segregate the activities of the more ancestral ligands of the superfamily from the more recently evolved. But obviously, there needs to be other mechanisms. I mean, there's more than just two ligands in the superfamily. So how are the activities of, for example, the individual TGF-betas discriminated from one another? And so we've been interested in trying to understand that as well. What's similar and what's different about how the three TGF-betas bind the TGF-beta receptors? If we look at those structurally, can we decipher any differences in the way they interact with the receptors that might account for the differences in biological activity between the three TGF-betas? We know that there are differences in the biological activity of the three TGF-betas. We know that because if you knock out any one of the TGF-betas in mice, just one by one, the mice are non-viable. So the three TGF-betas have unique roles in vivo, and we'd like to try to understand if that's due to differences in the way that they're binding the TGF beta receptors into a signaling complex. And the bottom line, and I won't go through the data because there's quite a bit of it, is that we do think that there are differences in the way in the three TGF betas and in the way that they bind the receptors. So I'm not going to, as I said, I don't have time to go through that. But let me just skip to the end. Let me skip to the end. Sorry, we'll be in here in a half hour. I just want to end here and just say that the work in my lab is largely done by PhD students. So the work on the first project that I spent essentially all the time telling you about was mainly done by this PhD student, Jorge Zuniga. He's actually Peruvian. Jorge got some help from a postdoc who came into my lab, TBRI. And then the project that I didn't have time to tell you about on the structural characterization of the three TGF betas and identification of differences, how they bind the receptors, was done by a more recent PhD student in my lab, Cao Huang. So the only reason I mention this is that, you know, if you're interested in research, this is the kind of depth in which you'll get into doing research as a PhD student. So all these students have multiple published papers, and they're all now gone on to postdoc positions, and they'll either get jobs in companies or in academia. And so I guess the last thing I want to say is if you are interested in graduate studies, talk to the faculty here at UPS. I think they can, there's a lot of graduate programs across the country. We have a graduate program in biophysics and biochemistry in San Antonio, but there are many good graduate programs across the country. The faculty here, I think, can advise you of ones that would be good to match your interests. And the other thing about graduate school is that in biomedical sciences, it's pretty much universally supported through stipends. And stipends are actually, I would say, especially in the past 10 years, significantly improved from what they have been in the past, because they really do want to encourage students to come into science. It's a lot of fun. It's a lot of work, but it's also a lot of fun. So I hope I conveyed that to you, and I'm more than happy to answer any questions about what I told you about. Thank you.